Good afternoon, everybody. So we'll just wait for 2-3 minutes for rest of the people to join, and then we will start the session. Okay. Okay, let us start the session. So a very good afternoon to everyone. Just a minute. Yeah, fine. So a very good afternoon to everyone. This is Purva Soni, and this is the last class of our live interactions session where we will cover the topics which were covered in week 12. Okay. So after this class, I think you will have your final examinations. You must have gotten your date for the final examinations. If not, then I think you will be getting it soon. But I think you have already received the dates. So here we'll be covering the 12th week lectures. But if you have any problems regarding any of the topics that we have covered in these 3 months, then you can always ask me. And like the YouTube page where I post all the recordings, that is also available to you. So if you can comment there also, if you have any query, any questions that you want to ask, so there also you can put it. So I hope that up till now, whatever we have covered, you were able to understand and grasp something out of it. This course, RNA biology, actually is an important topic in relation to the molecular biology subject, not only for this exam point of view, but as I always say, for your competitive examination point of view also. And if you want to further go for masters or PhD, even then, that time also, this RNA biology course, whatever contents that are there in this course, are very helpful for building your basic concepts, which can help you to do good in your studies as well as in your research. So let us start today's session. So lecture week 12 is about epitranscriptome and protein synthesis. So in this week we'll be talking about epitranscriptome and protein synthesis. So just always remember ohm. Whenever we use the term ohm, here we are saying epitranscriptome. Okay. So whenever we use ohm, this means that we are talking about the complete set in that particular organs. For example, if I say gene, it's one single gene I'm talking about. But when I say genome, so genome means the complete set of genes which are present in an organism. Okay. When I say metabolites, yeah, metabolome. If I say metabolome, then it means the complete um metaboloids which are present in an individual, that is metabolome. So wherever there is om, the suffix om is there, this means the complete set of that particular thing in that individual. Okay. So now comes what is the meaning of epitranscriptome. Okay. So transcript is, I think everybody knows by now. This is the last class of RNA biology. So transcript means uh, which has been synthesized from DNA. Epitranscriptome means, and what is synthesized from DNA? I mean, what is made after DNA? It is your RNA. So transcript is your RNA. That is fine. Now, epi word, which is extra over here, what does it signify? So the epitranscriptome includes all the biochemical modifications of the RNA within a cell. Biochemical modifications of the RNA within the cell. So what is epitranscriptome? All the biochemical modifications that are happening within a cell. The epitranscriptome refers to a variety of dynamic and reversible chemical modifications that affect the RNA transcript. That means whatever modifications that are happening, biochemical, chemical modifications that happen to the RNA that affects the RNA transcript, that is known as all the biomodifications that happens. So we are talking about the RNA, we are talking about the modifications, all the modifications. And these modifications can happen in the coding region of the RNA as well as in the non-coding regions of the RNA. So both coding and non-coding RNA chemical modifications complete together are known as epitranscriptome. So unlike the effects of DNA and histone modifications on gene expression that mainly occur at transcriptional level, okay, the effects of RNA modification on gene expression happen at post-transcriptional level. This is important. Happens at post-transcriptional levels, and RNA modification may affect RNA splicing, RNA stability, and RNA translation. As a result, RNA modification could upregulate or downregulate gene expression. This is important. That DNA modifications, whatever DNA modifications happens, that happens at the transcriptional level. But when we talk about epitranscriptome, which is all the RNA modifications which are happening, they happen at the post-transcriptional level. How we can say post-transcriptional level? Obviously, because RNA has been formed already. So transcription has already been completed. Transcription is completed. Now you have RNA. So whatever modifications are happening, or those are happening in the RNA. So obviously those modifications are at the post-transcriptional level. And RNA modifications, it can affect RNA splicing, RNA stability, RNA translation. So all these things can be affected by modifications of RNA. And as a result, RNA modification could upregulate or downregulate the gene expression. Okay. Now, this was just like a basic introduction that I wanted to give you about what epitranscriptome means. Okay. Now we will see, now we'll come to the lectures. So the lecture 1 in this week 12 was important RNA modifications. Okay, so what are the important RNA modifications that happens? Because here we are studying about epitranscriptome. So epitranscriptome means we are studying about the RNA modifications that are happening. So we should know what are the important RNA modifications that happens. Before that, just let us quickly recapitulate that we were studying about the telomeres and telomerase enzyme in our previous class. So what are telomerase? Telomerase are the enzymes which add the telomeres at the ends of the chromosomes. Okay, that is what telomerase means. Now, these telomerase can immortalize the cells. How we can say that these telomerase can immortalize the cells? Because telomerase can continuously add the telomeres at the end of the chromosome. So the cell will not die because it can keep on replicating. Because the reason, one of the major reasons that why a cell cannot keep on replicating is because that the telomeres are exhausted. After certain rounds of replication, the telomeres are exhausted. And because the telomeres are exhausted, the cells cannot undergo further rounds of cell division. But if there is a mechanism by which there's a continuous addition of telomeres at the end of the chromosome, then the cell can keep on replicating. That means the cell will become immortalize. So therefore, telomeres are the enzymes which can keep on adding the telomeres at the end of the chromosome, and therefore it can make the cell immortal. So therefore, telomeres can immortalize the cells. And then telomeres, and this is the example, you can just go through it, nothing much to explain over here. And telomerase can restore youthful phenotype in live tissue. What is the meaning of this? It means that, see, the cell ages. There is a concept of cell aging. One of the reasons of cell aging is because the telomeres get depleted after each subsequent round of cell division. Okay. So if there is a mechanism by which the telomeres are continuously replenished, then the youthfulness of the cell will remain because the cell will not get aged. So therefore, we can say that telomerase enzymes, it can restore the youthful phenotypes in live tissues. So normal human dermal fibroblasts were transfected and then grafted onto mouse skin. This is an experiment that has been done. And mice grafted with telomerase negative control exhibited phenotypic signs of senescence. But mouse which were grafted with telomerase positive cells, matlab, there were two mice. There were two sections of mice which were grafted. One section of mice were grafted with telomerase negative cells, and another section of mice were grafted with telomerase positive cells. So mice which were grafted with telomerase negative control cells, they had signs of senescence. And what are the signs of senescence that were observed? Increased fragility, reduced level of collagens, and subepidermal blistering. Whereas mice which had telomerase positive, which were grafted with telomerase positive cells, they exhibited a youthful phenotype, and they had, despite the same number of replications, because telomeres were continuously replenished. That was the reason. So this was just a recapitulation that telomerase can immortalize the cell, and it can add and it can restore the youthful phenotype in live tissues. Then telomerase can restore youthful phenotype in a whole organism also. This is also important. There has been a study in 2010 where showed that I'll just see, which showed that telomerase activation can dramatically reverse the effects of aging, and restoration of youthful phenotype was seen in testes, spleen, livers, intestines, and additionally effects of telomerase activation included, and not just that the morphologically you can see that there is a restoration of youthful phenotype, but also there was a restoration of fertility, reversal of cerebral atrophy, reactivation of neural progenitor cells. So these things were also there. Okay. And then telomerase effect activity predicts longevity. So it also predicts the longevity. The telomerase activity, how good telomerase activity is, that predicts the longevity. Okay. Then telomerase activators. Now here we come to one of the things by how telomerase can be activated. Telomerase have to be activated in a normal cell. What happens is telomerase is switched off. When I talk about normal cell, I am talking about somatic cells. Okay. In somatic cells, okay, and we have already seen that graph. I hope you remember that graph that we have discussed in our previous class, that with the number of replication, the activity telomerase numbers gets decreased. But if there is any sudden uh, activation, then again there can be an increase, and then the cell will become. So now this is how can telomeres be activated. Telomerase can be activated. So for there is a compound which is known as TA65, telomerase activation 65. It is the name of a compound. Okay, this is a compound. It's a molecule actually, which is derived from astragalus, and it activates the TRT gene, and therefore it activates the telomerase gene. And in vitro, it has been shown that it moderately activated telomerase, and in vivo also it has activated. So this enzyme activates telomerase. So TA65 activates the telomerase enzyme. And also, not just this is a compound externally, if this compound is given, then it will activate the telomerase activity. But also there is another thing that can activate the telomerase activity, which is exercise. So see, all the good habits and like excess drinking water, eating nutritious food, having a proper meal, and having a proper sleep and wake cycle, doing exercises, this is good for your body as a whole. Okay, each and everything of the body, like whether it's a proper functioning of our nervous system, whether it is the proper functioning of the reproductive system, or in this case, see, telomerase activity is very small molecular level thing, but each and everything, each and every biochemical reaction, and each and every physiological process that happens in our body is affected by our good habits, and it's affected by our daily lifestyle, putting it more correctly. So exercise, doing exercise will not only make keep you fit, but it will also increase the telomerase activity, which will keep you young. So therefore, and also we have seen in our last class, in a previous lecture, that stress, there are certain factors which can increase the depletion of telomeres. Okay, we have uh, talked about those factors also. So one of the major factors was stress, if you remember. So if you take stress, so stress is not good for your body in any ways. It's not only that you will get mentally stressed, and you will have you cannot focus, and you doesn't want to eat. But if you see on molecular level, so how those are the phenotypic changes or phenotypic reactions that you see, that you're unable to focus, unable to concentrate, you're feeling depressed, you are not eating proper meal. But molecular level inside, at a very minute scale, what is happening, some of the molecular changes are happening in our body. One of them is telomerase activity and telomeres depletion. So exercises also increases the telomerase activity. Now, RNA modifications. Now, this was just this much, whatever we have covered up till now. It was just a like the continuation of the telomerase topic that we were studying. Now, here in this class, we are studying actually about epitranscriptome. Epitranscriptome includes all the biological or biochemical or chemical RNA modifications that happens inside a cell. All the RNA modifications. So let us see one of the important RNA modification that happens, and that is N6-methyl adenosine. Okay. N6-methyl adenosine is an important RNA modification that happened, which is the most abundant modification. Okay. So what is this? N6-methyl adenosine, which in short we write as M6A. Okay, it is an abundant modification in mRNA and DNA. It is a dynamic and reversible process, and is the most pervasive modification in human mRNA. And it is found in some viruses and most eukaryotes, including mammals, insects, plants, and yeast. So it is not like it is only present in humans. No, it is present in most some viruses also. It is present in mammals, in insects, in plants, and yeast. Everywhere this M6A, which is M6A means that at the adenosine is added at the sixth position of the methyl. Okay. Then it is also found in tRNA, rRNA, and small nuclear RNA, as well as several long non-coding RNA such as ZIST. ZIST we have already studied, if you remember. Where we have studied ZIST? We have studied ZIST when we were studying the concept of dosage compensation, if you remember. Okay. This is a long non-coding RNA which helps in dosage compensation. Anyways, so this modification is not only observed in mRNA, but it is also observed in other forms of RNA such as tRNA, rRNA, and some small nuclear RNA, as well as this modification is also observed in several long non-coding RNA such as ZIST. Okay, then the methylation of adenosine is directed by a large M6 methyl transferase complex containing METTL3 as the SAM binding unit. Important point, I'm just marking it as important. And Okay. So what is this? The methylation of adenosine. So basically adenosine is methylated at the sixth position. And this methylation is carried out with the help of an complex which is known as M6A methyl transferase complex. Okay. And in vitro, this methyl transferase complex preferentially methylates the RNA oligonucleotides containing GGACU, and a similar preference was identified in vivo in mapped M6A sites in Rose sarcoma virus genomic RNA and in bovine prolactin mRNA. You can just, nothing much to explain, just you can go through it. Okay. Then N6 methyl. Now, this is an important slide. Here we will be covering two concepts, which is writer, readers, and erasers. Okay, now what is this concept of writers, erasers, and readers? So now M6A, this modification is divided into three types. Three kinds of modifications can happen. First is writer, second is eraser, and third is reader. Let us study as writer. Writers, uh, readers, and erasers. Okay. Writers means who writes something. Reader means who reads what has been written. And eraser means something that erase what has been written. Okay. So writers means those who add the modification. Readers means those things that read those modifications. And eraser means that erases those modifications. And which modification we are talking about? We're talking about N6 methyl adenosine modification. That means addition of the methyl group on adenosine. That modification we're talking about. So writers add the methyl group on adenosine and erases it, and readers reads it. So the modification is N6 methyl adenosine. Some molecules, some components will add the methyl group on the adenosine. Those will become writers. Some will read those modifications. That means recognize those modifications. When I'm talking about reading, that means recognizing those modifications. Okay. So some will recognize those modifications and those will become readers. And some will erase those modifications and that will become erasers. Okay. So more recent studies have characterized other key components of M6 methyl transferase writers. Okay. Hypothesis is that M6A in mRNA being dynamic and reversible is proven with this. Okay, just you have to understand that writers such as these are the examples of writer NPTEL 14 and all these, they will add the methyl group on the adenosine. Erasers such as FTO and ALKBH5, they will remove those modifications and readers will recognize the methylated adenosine on RNA. It will recognize the methylated adenosine on the RNA and that becomes your readers. Okay, so writers, readers, and erasers. So writer adds the methyl group on adenosines and erases it. And readers, proteins that recognize the modified adenosines. Okay, so I hope this difference of writers, readers, and erasers is clear to everybody. Okay, then next is, see, now this profile, this M6 methyl adenosine profile, this can be used to detect diseases. For example, this profile is a hallmark of cancer. That means M6A profile, it can be used to detect the cancerous conditions. Okay. Just wait for 1-2 minutes. Actually, this is lunch time, so I just ordered my food. So I'll just quickly go and take it from the delivery boy. Just 2 minutes, okay? Just 2 minutes. Okay, so now this M6A profile, it can be used to detect the cancerous conditions. Okay, and M6A methylation, it plays a important role in a wide variety of biological and pathological processes such as cancer development. And this METTL3, it is a methyl transferase, and this is upregulated in gastric, colorectal, and liver cancers. Okay, and this modifications are the key components of these pathways. So this is all, which means that the overall gist of all this is that the differential M6A profile is an emerging hallmark of cancer. It can be used to detect the cancerous conditions. How? Because whatever the modifications that happens at the M6A, that that modifications are frequently involved in some oncogenic pathways. Okay, so these modifications are frequently involved in oncogenic pathways such as Wnt beta catenin pathway, PI3K, AKT, mTOR signaling pathways. So these modifications are involved in oncogenic pathways. So now this is just an example, just to show that this is why HTDF1 driven gas translation of M6A. So why HTDF driven translation of M6A modified genes targets conferred malignant phenotypes using cancer cells' intrinsic properties such as proliferation, metastasis, stemness, and death. So you can see the different roles of YTHDF1 in gastrointestinal cancer. So if it gets methylated, see how many things, if this gets methylated and if there is an aberrant methylation, it can have effects on so many things. It can affect so many different pathways, which can lead to tumor genesis and metastasis, stemness, drug resistance, autophagy and ferroptosis, bacterial infection and inflammation, and immunity. So many things can be affected just because of this particular aberrant methylation that happens at the adenosine. So M6A methylation, modification can affect so many things. Then again, we come to lecture 3. Here, the biological implications of RNA modifications. So, up till now, we have studied that what kind of RNA modifications happen. The most abundant kind of RNA modification that happens is M6A. M6A is the most abundant modification that happens. Okay, this we have studied. Then we have studied that how this modification is regulated. So, how this modification is regulated? Because we have writers, readers, and erasers. And there are several examples of writers, readers, and erasers. And these modifications can alter the oncogenic pathways, and therefore it can lead to aberrant conditions such as cancerous conditions, and even one molecule if it is dysregulated. For example, we saw here only one, if it is dysregulated, you can see it can lead to so many different kind of phenotypic things. Now we'll see that okay, these modifications are happening, what are the biological implications? So this is the figure that describes the biological implications that happens. Okay, so I'll give you 2 minutes, just go through this figure and try to understand. This is a Wnt beta catenin pathway, so I hope this pathway is clear to everybody. Just go through this pathway and try to understand that how these biological implications happens. Okay. So have you gone through this diagram? So see what is there? This is the Wnt molecule. So this Wnt molecule, if Wnt, it can be studied in two ways: Wnt on and Wnt off. Okay. So Wnt on means when Wnt is present, and Wnt off means when Wnt is absent. So when Wnt is present, what happens is that beta catenin, it goes to the nucleus, and there is transcription of the Wnt target genes. Okay. Whereas when this Wnt is not present, then what happens? Then there is, if Wnt is not present, then there is destruction of beta catenin by its degradation complex. Okay. So you see, this is the ligand, Wnt ligand. It binds to its receptor. What is the name of the receptor? Frizzled receptor. When Wnt binds to its receptor, then disheveled is activated. And this disheveled inhibits GSK3 beta. Actually, this GSK3 beta is the one which kinds of phosphorylates the beta catenin, and therefore the beta catenin is marked for ubiquitination. But because GSK3 beta is inhibited, therefore beta catenin is not phosphorylated, and therefore beta catenin goes inside the nucleus, and then it activates the Wnt target genes. One of the Wnt target gene is MYC, and when this MYC, one of the this is Wnt target gene. Another thing is that when this YTHDF1 is translated, and if there is a modification then happens, then this also affects. So you see examples of YTHDF1 driven translation of M6A. Increased frizzled genes promoted the stability of beta catenin by the inhibition of GSK3 beta, and elevated TAS4 enhanced mediated transcription. Yes. Then c-MYC is an oncogenic target of Wnt, is also induced by YTH. So basically, so one of the target genes of Wnt is c-MYC, and c-MYC is an oncogenic target, and it is also activated by. This is the important point that you have to remember, that ex examples of YTHDF1 driven YHTDF1 driven translation of M6A modified gene targets include frizzled TCF4 and c-MYC. Okay, so I hope this is clear. Then now this is just for you to understand. I have just put some of the information. I'm not going to explain it all because it's a lot of information. I just wanted to understand that what this means. Okay. Just, oh, sorry. It's happened. Just for you to understand. This information is just for your understanding. Okay. Then we come to RNA translation. So basically, this RNA translation is an important topic here that we'll try to give much of our time in this RNA translation. But so basically, whatever translation happens, so there has been some information which has been given. This is all the information that has been provided to you. And okay. But we'll be dealing it like this. Hmm. Lecture 4 is epitranscriptome and protein synthesis. This is the information which has been given to you by sir. Okay. This is all the information given to you by sir. But I have made it in a different form where I have also included the information, but it is little bit more, it would be more clearer for you. Okay. So we'll do by this. Hmm. Okay. So now we have studied about what is epitranscriptome. Now we'll study about protein synthesis. Okay. So epitranscriptome, what is epitranscriptome? Then it includes all the biochemical or chemical RNA modifications that happens inside the cell. Therefore, we're talking about the post-transcriptional modifications. Whatever modifications that we talk up at the DNA level, whether they are DNA modifications or histone modifications, those modifications happen at the transcriptional level. But when we're talking about RNA modifications, these are the modifications that happens at the post-transcriptional level. Then we will study about translation. Now, we are studying because this is an RNA biology. Okay. So we'll be studying the role of RNA, the importance of translation in regards with RNA. Like what is the role of RNA in translation? So why we are studying translation? Because RNA, we are studying RNA biology, and RNAs are also involved in translation process. In fact, the ultimate goal of RNA is to serve the translation process only. There are three major types of RNA that we know. Okay, what are those three major types of RNA? These are mRNA, rRNA, and tRNA. mRNA is the messenger RNA, rRNA is the ribosomal RNA, and tRNA is the transfer RNA. And all these three RNA are involved in the translation process. How they are involved? mRNA specifies the amino acid sequence which codes for the protein. Which amino acid has to come in which order? Understand it, what I'm trying to say. Which amino acid has to come and in which order? That information is provided with the help of messenger RNA, and that is why it is known as messenger. It has the message which has to be delivered. Then we come to and how this information is read. So this whatever ATGC sequence is there, they are read in pairs of three. So you have three triplet, and each triplet is known as codon, and each codon specifies for one amino acid. So whatever is the sequence of the mRNA, this sequence of mRNA is read as triplets, and each triplet specifies for one codon. And in this way, sequentially, depending on the sequence of the mRNA, amino acids are added. Okay. So like this, mRNA helps in deciding the sequence, which amino acid has to come and the sequence of the amino acid out also. Then next is tRNA, which is the transfer RNA. Now, what is the function of transfer RNA? This transfer RNA, it converts the information in mRNA codons into amino acids. That means, okay, first mRNA is there. Now, ATG is there. Whatever is there, AUG is there. Now, it codes for methionine. So, now methionine has to be brought and added. Now, who will bring the methionine amino acid? That transfer RNA will do. So now whatever information is there, it will convert that information into amino acid, and it will carry that specific amino acid. So there it is transferring. By the name itself, you can see transfer RNA, it is transferring. So transfer RNA will transfer the information, and it will carry that particular amino acid that has been dictated by your mRNA. So tRNA, it will carry the amino acid specified by the codon. It will carry the amino acid which is specified by the codon. It will bring it to the mRNA. This is your transfer RNA. And this how it reads. It reads it via codon anticodon base pairing. Okay. Then we have ribosomal RNA. Basically, ribosomal RNA is the one which makes the peptide bond formation. So rRNA is required for translation. But what exactly it does is it catalyzes the peptide bond formation. Peptidyl transferase enzyme, it is present in it, that is a component of rRNA. So this ribosomal RNA, it catalyzes the so that means peptide bond formation happens because of ribosomal RNA. Okay, I hope this is clear. So thus from this slide, what we get to know is that during protein translation, our RNAs have a lot of role. So here we are studying basically the role of RNAs in protein translation. What is the role of RNA? So there are basically three types of RNA: mRNA, rRNA, and tRNA. And all these three types of RNA, they have different roles and they support the process. Actually, this whole translation happens because of RNA. Okay, the ultimate work of RNA that is formed after DNA is to carry out translation. Okay, then we have the genetic code with us. Now, what is the genetic code? This genetic code is important, and there are certain characteristics of genetic code that we will be studying later. Okay, then see now how we can read the triplets. So there are some important properties that you should remember, that these triplet codons are non-overlapping and commaless. Okay, so the important properties of a triplet is that these are non-overlapping and commaless. Okay, this is important, and they are read in the pair of three. This is frame 1, frame 2, frame 3, but in any frame they can be read. But in which frame they are read, uh, that frame reading will specify and it will make a different codon. Therefore, frame shifting is very uncommon here because it can specify a different codon altogether. So, frame shifting is a little uncommon, but this is generally just to tell you that what are the different frames in which it can be read. So, see, it can be 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3. This is one frame. Then you can leave one and you can take from second and 1 2 3 1 2 3 1 2 3, or you can leave all these two and then you can start 1 2 3 1 2 3. So there this what in which way it is read, it will create different polypeptide. So same mRNA can be read in more than one frame, but it is very rare. So what is written, that rarely are mRNAs read in more than one frame. So frame shifting is very uncommon. It is important. Frame shifting is very uncommon here. Then this charging of the mRNA, this is the figure that tells how the charging happens. This we will be studying. I have made some. This is the structure of RNA. This all we are going to study. Codon, anticodon, base pairing, ribosomes. Okay, so those are slides, just like the screenshots of, I mean, whatever has been given by the sir, and that is equally important. And you have to go through those slides, and therefore I have kept the screenshots just to make you understand in a simpler way. I have made this little bit by myself so that you can understand the process of translation. Okay. So the first thing is, translation in eukaryotes is the biological process by which protein is synthesized from the information coded on the molecule of mRNA. That means whatever information is given on the mRNA, whatever information is present in the mRNA, according to this information, your protein is synthesized. It can also be defined as the process in which nucleotides sequence on the mRNA molecule helps to incorporate amino acids into the proteins. Yes, exactly. So now, what are the components of a eukaryotic translation? So whenever we talk about the components, so there are different components. You have mRNA, tRNA, ribosomes, some transcription factors, and some enzymes. So mRNA, it contains the DNA sequence or the genetic information in the form of codons. So mRNA has the information, the genetic information in the form of codons. tRNA, it is like an adapter molecule. Adapter. So tRNA is an adapter molecule between amino acids or codons, which is required to carry the amino acids to the ribosomes. Then ribosomes, we are talking about eukaryotic. So we are talking about the ATS ribosomes. These are the protein synthesis machinery. It also has rRNA, which helps in translation. Then we have the transcription factors such as initiation, elongation, termination. And then majorly two kinds of enzymes are involved. First is amino acyl tRNA synthesis, and second is peptidal transferase. So basically, what does this amino acyl tRNA synthesis does? It catalyzes the attachment of tRNA to its amino acid. Okay. So basically, the attachment of tRNA to its amino acid is catalyzed by amino acyl tRNA synthesis. And second is peptidal transferase. Now, what's the function of peptidal transferase? Basically, it catalyzes the peptide bond formation. So these are the components of a eukaryotic translation. And this we have already studied. Okay. Now the genetic code. Now these are the properties of genetic code. This is an important slide. Okay, so what are the properties of genetic code? They are mostly triplicode triplet. So these are triplets. They are unambiguous and very universal in nature. They have a degenerate code. What is the meaning of degenerate? It implies that one amino acid can be coded by more than one codon. Okay, I'm writing it here. Degeneracy means but this is an important thing that I have written, that one amino acid can be coded by more than one codon, but one codon can code only for one amino acid. Just read this line. I'm giving you 1 minute. Read and go through this line so that you can understand what I'm trying to say, what I'm trying to tell. Go through it. Did you understand what I'm trying to tell from this thing? So which this means that suppose if AUG is the codon which codes for methionine. Let's say methionine can be coded by some other sets of codons also, but this AUG can code only for methionine. So that is what it means, that one amino acid can be coded by more than one codon. That means one amino acid can have more than one codons, but this codon will only code for methionine. So that is, they have a degenerate code. They contain start and stop codon. So mostly the start codon is methionine AUG. They showcase polarity, 5' to 3' polarity. Their code is mostly non-overlapping and there are commaless, hence have no indication of an end or a beginning. Okay, so these are the properties of genetic code. And then there is codon anticodon base pairing. So what happens is that you have to just go through it. The third position is called as a wobble base. Okay, so base pairing between the third position of the codon, this is the codon, third position of the codon, and first position of the anticodon is known as a wobble position, and it is less constrained, and therefore it leads to the different kinds of diversity. So wobble position is important. This is basically first position of the anticodon and third position of the codon. Okay, then we come to charging of tRNA. Now, this is an important slide. So, what is the meaning of charging of tRNA? Charging of tRNA means that this tRNA is an adapter molecule which brings the amino acid. Okay, now how it will bring the amino acid? So, first is that this will bring the amino acid with the help of some ATP energy requirement. So first of all, whichever acid has to be added, that amino acid is activated, and amino acid is activated by the attachment of ATP, and then that activated amino acid binds to the tRNA. So whichever amino acid has to come and it has to bind to the tRNA, it has to be activated. Then only that amino acid can bind to the tRNA. And that activated amino acid, when it binds, and then it becomes amino acyl AMP. And this amino acyl AMP then goes and bind to the tRNA and it is formed amino acyl tRNA. So this, you see here, this is amino acid and there is ATP, and amino acyl tRNA synthesis enzyme which is responsible here, amino acyl tRNA synthetase. So what happens first, there is activation of amino acid. So now you have amino acyl AMP. This amino acyl AMP is the activated amino acid. Okay, now this amino acyl AMP, which is the activated amino acid, will go and attach to the tRNA, and when this activated amino acid, which is an amino acyl AMP, go and attaches to the tRNA, now this tRNA will be called as amino acyl tRNA. Amino acyl tRNA, which has the activated amino acid attached to it, that is your amino acyl tRNA. So I hope this charging of tRNA is clear. Charging means that amino acid is attaching to the mRNA, to the tRNA, but not any random amino acid can come and attach. First, the amino acid has to be charged. That means the amino acid has to bind with ATP, and then this charged amino acid will come and bind to the tRNA. So this is your charging of the tRNA. Then we come to the mechanism of translation. There are three steps, like we have studied in transcription also. There is initiation, elongation, and termination. Similarly, in translation also we have three steps: initiation of translation, elongation of translation, and finally termination of translation. Okay, So initiation of translation means the first uh, uh, initiation of translation means that the small ribosomal unit and large ribosomal unit will come and bind with each other, because generally when translation does not happen, the small and the larger ribosomal units are not together. They are apart. They are away from each other. Only when translation happens, the small and the large ribosomal units come and they get bind, and then first tRNA comes and it attaches to the P side. And the first tRNA is the initiator tRNA, which brings methionine generally. Okay. So you see, this is the small ribosomal unit, and this is the initiator tRNA, which has methionine attached to it. And this is the start codon. This will come, it will enter from the 5' end, and then it will scan where the start codon is there. It comes, it scans where the start codon is. It got the start codon, it gets attached to it. After this, the large ribosomal unit will come and join. So first, initiator tRNA comes, scans the start codon, gets attached, and then finally the large ribosomal subunit joins and initiation complexes form. Okay, then this is transcriptional initiation. We're talking about this is in eukaryotes. Okay, this is eukaryotic translation initiation. So what happens? This is the 40S ribosomal unit. This 40S is bind to the factors such as eukaryotic initiation factor 3 and eukaryotic initiation factor 1A. And then comes and meets the methionine tRNA, and then it forms the 43S, which is the pre-initiation complex. This 43S, then 60S RNA will come and it will form uh, sorry, this 43S will bring the mRNA and then will form the 48S initiation complex, and then the larger subunit comes and bind and it becomes the complete initiation complex. You can just go through the diagram. And then we come to transcription elongation. So what happens, that in ribosome there are 3 sites. Okay, this is the ribosome. There are 3 sites which are important: E, P, A. E is the site where the first tRNA, which is the initiator tRNA, it brings methionine. Okay. Okay. And then, so first tRNA, which has the initiate, which is known as the initiated tRNA, it brings the first amino acid, which is the methionine, and it comes and it binds to the P site. Subsequent, all the tRNAs which brings the subsequent amino acids, they come and they bind to the A site. Okay, so first tRNA comes to the P site, and second comes to the A site. So second and all the other, it comes to the A site. So there are three major steps in translation elongation. First is binding of amino acid tRNA to the A site, peptide bond formation, and translocation. So elongation means that the amino acid tRNA will come, it will bind to the A site, then there would be peptide bond formation, and then there would be elongation, translocation. So what happens that the P site has the methionine tRNA and the A site is free. So what happens that the next first amino acid comes and it binds to the P site, and then the next amino acid comes and it binds to the A site, and then there is peptide bond formation happens. This peptide bond formation is catalyzed by the enzyme peptidyl transferase, which is the component of rRNA. Okay, peptidyl is a component of the RNA, and then there would be translocation. It will move. So what happens? This will move like this. See, this is moving like this. Ribosome will move towards the 3' end. So what happens? This ribosome moved. So now this P site will come to the E site, A site will come to the P site. So E means exit. Now this, which is actually empty, will be get will exit, and now there are two bonds here. Okay, this is the first amino acid, and it bonded, and it, see, this forms a peptide bond. Now this shifts here like this, like this is shift here. So A comes to P, P comes to E, like this ribosomes move. Then we come to the termination of translation. So there are two stages in the termination of translation. The first is the cleavage of the growing chain, and the second one is the recycling. Okay, so what is cleavage of the growing chain? So one of the stop codons. So how this happens is just hmm. So for the termination of translation, you have three stop codons, okay, which gives the termination signal, which is UAA, UAG, and UGA. And these stop codons are recognized by some release factors. Okay, so eukaryotic release factor 1 helps in recognizing, it recognizes the stop codons. And eukaryotic release factor 3, it helps to stimulate termination. Okay, and then what happens? So what this is just like, okay, that there are three stop codons, and then there's a release factor 1, release factor 3, all this is fine. But actually what happens is that when the ribosome comes in contact with the stop codon, there will be no tRNA available for binding to the A site, and instead it will release, instead of it, a release factor will bind. So what happens? Ki whenever there is a normal codon, so when a ribosome comes in contact with normal codon, according to whatever codon, whatever amino acid that codon specifies, that tRNA will bring that particular amino acid. But when ribosome recognizes a stop codon, what happens is that instead of a tRNA, release factor will come and bind to it. Okay, this is important. So what happens that when a ribosome comes in contact with stop codon, then there is no tRNA that will come and bind to the A site. Instead, a release factor will come and it will bind to it. And after the binding of the release factor, what happens? The ribosome will fall apart into the following components. What are the four following components? Small and large ribosomal units will be released. Peptide product will be freed and transfer RNA will also be released. Okay. So this is what happens when a ribosome recognizes or encounters a stop codon. So instead of the transfer RNA, instead of a charged tRNA, then release factor will come and it will bind to it. Fine. And then there are two processes. First we have studied, second is the recycling. What was the first one? Cleavage of the growing chain. So the growing chain has been cleaved. Second is recycling. So what is recycling? Recycling of ribosome occur in eukaryotes. When the polypeptide and release factors are released, the ribosomes are still bound to the mRNA and have the deacetylated transfer RNA. So now the mRNAs and tRNAs which are there, they are not used for just one single round of polypeptide synthesis. No, they are reused. So therefore, for the new polypeptide synthesis, these mRNA and tRNA must be separated and ribosome must also be dissociated. All of these processes are collectively known as ribosome recycling. So in this way, ribosome recycling happens. Okay. So with this, we have completed the theory part. Okay. Now we'll do the questions. So question number 1 is, YTH domain proteins are considered as RNA modifiers, epitranscriptome readers, epitranscriptome writers, or epitranscriptome suppressors. So what is the correct answer? These YTH proteins, what are they? Modifiers, epitranscriptome readers, writers, what? 2 minutes and then I will switch the screen. Think what can be the answer? Mm. 1 minute more. Okay. So The correct answer is epitranscriptome readers. So this we have video. So YTH domain proteins we have already studied here. If you can see, hmm, this YTH domain family have been characterized as direct MCA readers and have a conserved M6A binding pocket. So this question which says that YTH domain proteins are considered, so they are considered as epitranscriptome readers. Okay, fine. Next question is, which of the following is a frequent RNA modification? Deadenylation, N6-methyl adenylation, ubiquitination, and summarization. So we have to tell which is a frequent RNA modification. Something that we have been studying since the beginning of this class. Okay. So try to answer this question. In 2 minutes, I will switch the screen. Okay.
इसके डिटर्जेंट को खत्म करने के लिए कुछ डालना है पानी से
One more minute left. Think. So the correct answer is N6-methyl adenylation. So all the time we have been studying that N6-methyl adenylation is a RNA modification which is regulated by three components: writers, readers, and erasers. And this RNA modification has, if this RNA modification is dysregulated, it can have many aberrant effects on many of the oncogenic pathways, such as Wnt beta catenin pathway, TNF pathway, and etc. And this can lead to several cancerous conditions. Okay, so the correct answer is N6 methyl adenylation. Okay, next question is, factors that cause an RNA modification is called readers, erasers, writers, or modifiers. So we've already studied about what are readers, what are erasers, and what are writers. See the question carefully. They're asking factors that cause the modification. So think you have 2 minutes to answer this question, and then I will tell the answer. One more minute left. So the correct answer is writers, because writer will add the methyl group to the adenosine residues. That means they are causing the RNA modification. Readers will recognize that modification, and eraser will erase the modification. The factor which will cause the modification, that means which is writing that modification, is writers. So that is the difference between writers, erasers, and readers. Writers will add the methyl group to the adenosine residues. Readers will read that methyl modification, and eraser will erase that modification. There is nothing like modifiers. Okay. Then next question is, bacterial mRNA codons are A, non-similar to plant codons, B, similar to other animal codons, C, not similar to virus codons, or D, similar to the mitochondrial codons. What is the answer? One more minute left. Think what can be the answer. Okay. So the correct answer is similar to other codons. So why we have said when we were studying the properties of codons, what we have studied the property was it is universal. That is codons are conserved and they are very much universal. So their codons would be similar to the bacterial codons would be similar to animal codons. Okay. Then next question is before participation in protein synthesis, the tRNA must get modified, amino acylated, denatured, or trans-acetylated. Think. Okay, 2 minutes are over. Let us see what is the correct answer. So the question is, before participation in protein synthesis, the tRNA must get, so what it should get? It should get amino acylated. So we have studied in detail about the charging of tRNA. What is the meaning of charging of tRNA? Amino acid, whichever amino acid that has to be attached to the tRNA, first it should be charged. And how the amino acid will get charged? By binding with ATP. So amino acid will bind to the ATP and it will get amino acyl AMP. And then this amino acyl AMP will come and it will bind to the tRNA. And then it will become amino acyl tRNA. So this is known as charging of the tRNA. Okay. Just a minute. Okay. Therefore, the correct answer would be trans-acetylated. Okay, fine. Next question is mRNA codons are usually discontinuous manner, overlapping manner, three frames manner, or a distinct manner. Hmm. We have studied the properties of codon, so accordingly, try to answer this question. You have 2 minutes to answer this question. Okay, thank you. One more minute left. Okay, so the question is mRNA codons are usually discontinuous, overlapping, three frames, or distinct manner. Okay, and the correct answer is distinct manner. So, are they overlapping? No. We have already studied the properties of codons and we have studied that they are non-overlapping, they are continuous, and mostly one of the frame is only used. It is not like sometimes one frame is used, other time other frame will be used. So the changing of the frame is a very rare event when it comes to mRNA codons. So that means what is the correct answer? Here you have to select distinct manner as the correct answer because they are not discontinuous, they are continuous in fashion, they are non-overlapping. And although mRNAs can be read in three different frames, there is only always one frame that is being used and the frames are not changed. So frame shifting is a very rare event when we do in mRNAs. There is no shifting of the frames. Then protein synthesis can happen only from tRNA. True or false? Think. Can protein synthesis happen only from tRNA or there are other sources also? Think you have 2 minutes to answer this question, okay? One more minute left. Okay. So the correct answer is false because we have studied in the very beginning what is the role of RNAs in protein synthesis. Okay, so we have studied that there are three different RNAs: rRNA, mRNA, and tRNA. And all the three types of RNAs have their role in protein synthesis. All the three RNAs have their role in protein synthesis. So, ribosomal RNA, mRNA will provide the triplet codon. Ribosomal RNA will catalyze the peptide bond formation. And tRNA acts as an adapter molecule which bring the amino acid which is specified. So, therefore, protein synthesis can happen only from tRNA. It is a false statement. Okay. Next question is, adequate vitamin C in diet ensure longer telomere length. True or false? Think what can be the answer. Okay. So the correct answer is true. So we've already studied that all the good habits, all the good things that we do, like exercising and taking good nutritious foods, they all help in ensuring the telomere length. So the correct answer would be true. Next question is, mutations could be detected and eliminated from cells. True or false? So can we detect and eliminate the mutations? That is the question. think what can be the correct answer? Okay. So the correct answer is true. That mutations could be detected and eliminated from the cell. So basically, the nature has made itself in such a way that it tries to detect and eliminate most of the mutations that happens in the body. So it tries to eliminate most of the mutations, and therefore most of the mutations happens in the non-coding regions. So this is important that most of the mutations that happen in the non-coding regions of the DNA so that they can be easily detected and eliminated. So the correct answer is true. Next is low mental or physical stress maintain long telomeres. True or false? Again, very similar question that we have already dealt. One more minute left. Okay. So again, it is a true statement. We have studied that, see, lifestyle, these lifestyle changes It can help in maintaining long telomeres. Okay, and what are the lifestyle changes that I'm talking about? Exercise, nutrition, which includes the vitamin C wala concept also. The physical activity, it will come under exercise only. So physical and mental health. All this will help in maintaining long telomeres. Okay. So in general, you need to understand that whatever we are doing, what kind of lifestyle we have, what kind of systematically we are living our life, it affects our whole personality. In one molecular level, it affects our telomeres also. So this is the correct answer. So the correct answer is true. Okay. So just a minute. Hmm. So I think it should be clear now. Let us quickly revise what we have studied, and then we can just wind up the class. So we were studying about epitranscriptome and protein synthesis. So we have studied that uh, just a minute. Hmm. Hmm. So we have studied about epitranscriptome and protein synthesis. So what is epitranscriptome? Epitranscriptome includes all the biochemical modifications of RNA that happens in the cell. That is your epitranscriptome. Just a minute. Oh, what has happened? Okay, not presenting the screen. I'm sorry. Yeah. Hmm. So epitranscriptome, it includes all the biochemical modifications that happens inside the cells, and these modification can affect RNA splicing, RNA stability, and RNA translation as well. So then we have studied a little bit about the telomeres. We were continuing our discussion of the telomeres from the last class, that how telomeres can immortalize the cell and how telomeres can restore the youthful phenotype in the tissues, and then how telomeres can restore youthful phenotype not in a particular cell, but it can also restore it in the whole organism, and it can also restore fertility, reversal of cerebral atrophy, and reactivation of neural progenitor cells. And then how telomeres affect the longevity of a cell. Then we started with a component which is known as Ta65. So Ta65 is the component which is derived from Chinese herb which is known as astragalus, and this is required for telomeres activation. So with the help of Ta65 compound, we can activate the telomeres, or by doing some kinds of physical exercises, or by drinking good water, taking a lot of water, having nutritious food which is rich in vitamin C. All these things can also increase the telomerase activity. Then we were talking about RNA modification. So one of the most important RNA and the most prevalent abundant modification in mRNA is N6 methyl adenosine, M6A modification, and this modification is divided into three types: writers, erasers, and readers. So writer are the factors that will add the methyl group on the adenosines, reader which will recognize the methyl group and read it, and eraser that will erase that modification. Okay, so writers, it will add the methyl group on adenosine, erasers will erase it, and readers are the factors that recognizes those modified adenosines. Okay, then we come that these writers, erasers, and readers, their regulation is a hallmark of cancer. And if there is any kind of dysregulation or aberrant deposition, it can have role in various kinds of cancer by altering the gene expression. Okay. Then we studied about the examples of YHTDF, how it causes and how the modification is driven by YTHDF1. Then we talked about translation. So I will start the translation wala part here. Translation was in next this thing, no? Yes, yeah, sure. Hmm. So we talked about that there are all the three RNAs, mRNA, rRNA, and tRNA. All the three RNAs have roles in translation. So mRNA basically specifies the amino acid sequence of the protein, and your tRNA, it converts the information from the mRNA codons into amino acid sequence. It is actually an adapter, and your rRNA is required for the peptide bond synthesis. Then the genetic code, the properties of the genetic code. Before coming to the properties of the genetic code, we studied that the triplet code is non-overlapping and commaless, and although it can be read in three different frames, but mRNAs are usually read in only one frame, and the frame shifting is very, very uncommon in mRNAs. Then, sorry. Hmm. So we studied what are the different components which are required for the eukaryotic translation to happen, and then what is the structure of tRNA. So there is a so tRNA structure, basically its secondary structure and tertiary structure of tRNAs are different. Secondary structure of tRNA is a clover leaf structure, whereas tertiary structure of tRNA is an L-shaped structure. Okay. Then the genetic code. What are the properties of genetic code? They are triplet, they are degenerate, they have start codon and stop codons, they show polarity, they are non-overlapping, it is important, and they are commoners. Okay. Then we have said about the codon anticodon base pairing and what is the vowel position, then how the charging of the tRNA takes place, and then we started with the mechanism of translation, how translation takes place. So initiation, elongation, and termination are the three steps of translation. So how translation initiation takes place, how translation, what are the factors that are involved in the initiation of translation, what are the factors that are involved in the initiation of translation, then translation elongation. So basically translation elongation is divided into three steps: binding of amino acid tRNA to the A site, peptide bond formation, and translocation. So these are the three steps of translation elongation. And then in detail we have studied about translation elongation, and then we have studied about translation termination, how the termination of translation takes place. There are two steps in the termination of translation. The first one is the cleavage of a growing chain, and the second one is the recycling. So what happens, this point is important, which we have studied, that when the ribosome comes in contact with a stop codon, there will be no tRNA available to the binding to the A side, and what will happen, instead of a tRNA which has the amino acid sequence, what will come, a release factor will come and it will bind to it. Okay, release factor will come and it will bind to it, and after the binding of the release factor, the ribosome will fall apart into the following components. And what are the following components in which the ribosome will fall apart? It will fall apart into small and large ribosomal units. It means the small and the large ribosomal units will be released. The peptide product will be freed, and transfer RNA will also be released. So these are the different components in which the ribosome will fall apart after the cleavage of the growing polypeptide chain. So here we are talking about translation termination. So the first thing in translation termination is the cleavage of a growing chain. The second is recycling. What is recycling? So basically this recycling, it occurs in eukaryotes, and when the polypeptide and release factors are released, what happens? The ribosomes are still bound to the mRNA and have the deacetylated transfer RNA and the PNU sites. For the new polypeptide synthesis, these mRNA and tRNA must be separated and ribosome must be dissociated. This is an important point. that these mRNA and tRNA must be separated and the ribosome must also be dissociated. All of these processes are collectively known as ribosome reset. Yes. And then just look at this figure, see how what happens. This is the growing polypeptide chain, and here if it recognizes the stop codon, then instead of a tRNA, what comes is a release factor. And when the release factor comes, then everything just falls apart. This goes, the polypeptide is free, everything is just broken. See, this is the see see this figure for 2 minutes. So I hope this is clear to everyone. By this we have actually completed, and so now we have completed everything. So this figure, I think I couldn't give much time to it. So mm, yes. So see, this is structure of RNA. So primary structure is just the simply the sequence of the amino acids. Then secondary and tertiary structures are important. Secondary structure is a clover leaf structure. You can see this is a clover leaf structure, and the tertiary structure is a L-shaped structure. Okay, so you just see here, this is an important, it is secondary structure, and this is the tertiary structure. Just look at this structure. Hmm. This is the 5' end and this is the 3' end. The 3' end always have this CCA end, remember. Okay, 3' end has the CCA end. Then you have an acceptor arm, a DR, this is the T loop, this is the variable loop, and this is the anticodon loop. So this is the mRNA, and mRNA has the codon, and just the complementary to it, the tRNA has the anticodon. So this is the, so base pairing happens between the codon and the anticodon. Okay, so this is the anticodon, these are the codons. So these are the various, this structure is actually important. Where CCA is present, it is present at the 3' end. This is important, and this particular question is also important. Okay. So it's a clover leaf structure. Clover leaf. And this is the L-shaped structure. Okay. So this secondary structure, which is a clover-leaf structure, is actually important because it is frequently asked in examinations. This structure, when it is three-dimensionally, we will see, then you can see this is the CCA end, this is the acceptor arm, T loop, anticodon loop, and these are the codons, anticodons, variable loop, and T. So this is where it forms the secondary structure and the tertiary structure. So with this, we have completed the week 12th lecture. So I hope that things are clear to you. Does anybody have any problem? Any problems anywhere where you could not understand? You can tell me. Is it clear to everybody? So, if you have any problems, you can always comment on my YouTube channel. This video will also be available for you if you want to see the offline recording. You can see that also. And with this, we have completed all the 12 week lectures and all the classes. So we have started from RNA, how the RNA has evolved. We have studied about RNA world, how the RNA world hypothesis came, and then we have studied the different, what are the various kinds of RNAs, what is transcription, how transcription takes place, what are the different post-transcriptional modifications that takes place. We have elaborately studied about all the post-transcription modifications, and then we have studied about some different kinds of modifications that happens, which are alternative processing. Alternative processing was something which was new that we have studied. We all know, although we have studied in detail about splicing, polyadenylation, and all those things. But the alternative processing was a new part where we have studied about alternative splicing and how different poly A sites within the mRNA can be used to yield different polypeptide products. And then we have studied about RNA editing, how RNA editing takes place. We have also studied about non-coding RNAs and their roles, and how this miRNA is formed and how its biogenesis and how it controls the various regulations and all we have studied. We have also studied about dosage compensation and how dosage compensation is important. What is the role of non-coding RNAs in dosage compensation? We have studied about telomeres, how telomeres are important, and what is the enzyme that control telomeres. It is telomerase that we have studied. All that particular concepts we have studied. We have also studied about the epitranscriptome and protein synthesis, that even in protein synthesis there is a role of RNA, that we have studied. That was the main thing to study, that even in RNA, there is a role of that. Even in protein synthesis, there is a role of RNA. And epitranscriptome is also a important and a different topic that we have studied this time. Okay, so I hope this much is clear to everybody. And if you have any problems, any query, you can always come and talk to me and contact me. And now I think now you have your examinations. So I wish you all the very best for your examinations. Hope you do well. And yeah, that's it. Good luck for the future. And bye. It was nice teaching you all. So bye everybody. Hope you also enjoyed, and you also had a great time here. It's always fun to teach, although it's a part of our TAC work, but it's always fun to teach and interact with the new generation. So I think with this we'll end this complete RNA biology course. Not just the class, the complete course. So bye everybody.